Good morning. So let's continue on in today's lecture, discussing the process of translation. So how your cells take the edited mRNA message and converts that into a string of amino acids to make a polypeptide chain that then can fold up and represent a completed protein. And we'll take a look at the ribosome, how the ribosomes involved in that, and also the roles of the ribosomal RNAs and the tRNAs in helping to assemble that chain of amino acids. And then we'll end up today, hopefully, if we have time, looking at some of the consequences of different types of mutations on these processes. All right. So to review, the central dogma or central paradigm of information flow in living cells goes through DNA as the starting molecule of information, through an intermediate molecule, RNA, right, and then eventually into the creation of proteins. And we said last time that if you just look at this picture, this represents 2 out of the 4 main classes of macromolecules that we talk about in this class. The nucleotides that form the nucleic acids like DNA and RNA, right? And then amino acids that come together to form proteins. But then we said last time, well, what about things like lipids or carbohydrates? Because all 4 of those macromolecule groups, nucleic acids, proteins, carbohydrates, and lipids are vital for the functioning of the cell. But what I asked you to think about last time is that the actual information in our DNA, in our genes, right, is mainly concerned with just the creation of proteins, the set of instructions, which are our separate genes, to make different proteins. And then it's up to the proteins to function as structural molecules or little signaling molecules, or importantly, as little machines that will then coordinate, right, what to do with sugars and lipids that are in our tissues already, or the sugars and lipids that we get in from our diet. All right, so that's why proteins are really so important, because in many ways, proteins are what coordinate the other macromolecules, right? Not only the carbohydrates and the lipids, but of course, the nucleic acids themselves. As we've been learning, the process of organizing our DNA is a function of proteins working on them, wrapping them up in our chromatin. Also, those big complexes like RNA or DNA polymerase that come and copy DNA is a big group of proteins working together as a big machine, right? And that's the process of transcription. Right? So today we're also going to look at then the process of translation. When you have the RNA, you're going to have another big machine made up of proteins that's going to then translate that mRNA into making more proteins. Right? So recall that the intermediate molecules, the RNAs, there are 3 main types that we are concerned with. The mRNA, or the finished messenger RNA, which represents the original perfect copy of the gene as the primary transcript that then has been edited, right? Parts of the primary transcript can be cut out, other parts kept, and you get an mRNA or messenger RNA final version of it, right? Which represents the genetic code. Every 3 bases of that RNA stands for the position of where an amino acid should be in a chain of amino acids, right? So that messenger RNA contains the actual genetic code that's going to be translated into making a protein. The tRNAs and the rRNAs, right, they just function as being little RNAs that their structure helps contribute to the process of this translation taking place. Right? And so they are involved with the ribosome. Right? So again, over here, right, the process of reading the DNA in the gene to make RNA first as a primary transcript, and then finally editing it to form your finished mRNA, right? That would be the process of transcription. And so then translation is what we're going to focus on today, right? How your cells read that mRNA message and make sense of putting them together using that table of triplet codons that we looked at last time, right? So AUG codes for methionine, GGC codes for glycine, UCC codes for serine, right? So what is the machine that actually does this interpretation and the assembly? So that is the ribosome, right? So the ribosome is a great example of one of these big complex machines in your cell that's made of a combination of proteins, right? So the purple things in this ribosome are different protein subunits. And as we looked at last time, the squiggly brown things are different rRNAs that contribute to the correct shape of this ribosome to together make this factory. And then the mRNA is going to be fed through this. And what we'll look at today is that the tRNAs are going to be bringing individual amino acids, the methionine, the glycine, the serine, right, and linking them together inside this ribosome based upon the ribosome reading that codon sequence. Right. So ribosomes, again, are made up of proteins and rRNA subunits that come together in this big complex. Here's an electron micrograph of them. So these are large protein complexes. So these are the things on the previous slide that was just kind of rotating around. Right? In a eukaryotic ribosome, there are two main parts to it: a 60S large subunit and a 40S small subunit. Okay? Each one of these is made up of a combination of proteins and rRNAs. The S, what does that mean? That's kind of an old, antiquated term. It's a sedimentation rate. So back in the old days, when researchers were first beginning to study, right, what makes up a ribosome, they would take a whole bunch of ribosomes, disassemble them, right, so that they fall apart, and then they'd, in a test tube, spin them in a centrifuge so that the heavier components would end up going further down the tube, the lighter components would stay up. And so that rate of which these different components would form sedimentary layers within the spun tube, they use that as a reference for what these different units are. So that's where the S comes from. So as a result, S is not the same thing as strictly like size, because a lot of the migration or the sedimentation also has to do with the shape of these molecules. Things that are bunched up tighter, might migrate more than things that are maybe loose, right? So that's why if you say that this 60S large subunit and you have a 40S subunit, when you add them together, it doesn't become a 100S, right? It actually together migrates at 80. So together it's called the 80S intact ribosome. But each one of these, the large subunit and the small subunit, is made of a combination of proteins. So you can see the 60S is made up of 49 separate little protein subunits, and then these small rRNA components that we've talked about. We looked previously at how our cells are always making a ton of these small rRNAs from those tandem repeat regions of certain genes where transcripts are made at one time that contain 58S, 18S, 28S, and then those smaller parts are punched out to give you these separate little pieces, right? So that's the intact ribosome in our cells, eukaryotic cells. So just a quick note, prokaryotic ribosomes, things like in bacteria, it's a similar theme, right? But the component sizes are different because the actual proteins, the bacterial versions of these proteins, the more primitive ones, the sizes are a little different. So I point this out only because I think the book illustrations are a little sloppy. They kind of give examples of both eukaryotic and prokaryotic. So don't be confused if the whole like S size thing, doesn't line up right. Okay. So let's look at the anatomy of what's going on inside this ribosome. So in this big, massive protein factory, if you were to take a peek inside of this factory and actually see sort of like the work areas where things are being made, right? First off, these dotted lines that go through in between the 60S and the 40S, this would be sort of like a channel where the mRNA is going to be scanned through here. Or more accurately, the whole ribosome itself is going to be moving along the strand of the mRNA, reading it 3 bases at a time. Every 3 bases, not surprisingly, the space of every 3 bases fits within one of these little chambers here. This A site, which we call the amino acyl site, this is where a tRNA is going to bring the next amino acid into the ribosome. Right? And there's going to be this mRNA that's already kind of being read by the ribosome, and the 3 codon bases of the mRNA, which would be where this dotted line are going to dictate which tRNA is allowed to come in and properly bind there. Right? And that tRNA is going to be bringing the amino acid that corresponds to those 3, that triplicate code that you get from the table. The P site would be where the previous tRNA that was in the A site would then move over to the P site. This P site then would be where the previous amino acids that were brought in have already been linked together, right? And so the tRNA that's going to be sitting in the P site is going to be hanging on to this growing chain of amino acids, including the one that it just brought in when it first came in from the A site, right? So that amino acid that it brought in is going to be added to whatever chain is being held onto in the P site. Okay? And then the tRNA will move over to the E site, which is just the exit site, and then that's where that tRNA will leave. And there'll be a continuous series of tRNAs coming in here. So this is what it looks like. You know, I always love these more accurate 3-dimensional figures because even though the little cartoony blob thing here gets the point across, I think all of you are probably more interested in seeing what does it really look like, right? So here it is, the 60S up here is in the gray and the navy blue. So that's the combination of proteins in the sort of grayish areas. And then the navy blue are those different rRNAs that make up the 60S part. And then the purple ones down here are the 40S part. Right? They don't show the mRNA going through the middle here, but essentially you can imagine the dotted line over here, right? It would be somewhere going through the middle, but you can see a couple of the tRNAs that are in here. So this is little part peeking out here is part of the tRNA that's come in to the A site, right? The P site would be in the middle. It's blocked by the other parts of the ribosome here, but you can see the tRNA that's now in the exit site getting ready to leave, right? So if you look inside, this is what you would see. Here's the A site tRNA. Notice these little purple dots here are the individual amino acids, right, that are being carried in by the tRNAs and then added to the chain. So this A site tRNA that came in, it would be holding on to this little pink dot. That's what it brought in, right? The tRNA that came in before it has already moved over to the P site, and it is hanging on to this long chain of amino acids, right, including the one that it brought in, which would be the second one here. So what has happened is the long chain of amino acids that was held onto by the P site is transferred over to the amino acid that's being held onto by the A site. And that's how this amino acid gets added on. So it's kind of weird, right? It's not like you have a chain and the next amino acid gets sort of put on the chain. It's more like the chain itself gets put onto the next amino acid that comes in. It's more it's easier to think about it that way, right? Once the tRNA that was in the P site has passed the chain over, right, it moves over. Now it no longer has any amino acid on it. It can exit, right? So these tRNAs that are bringing in these little teeny tiny amino acids, right? Let's take a look at how these tRNAs do it and how the tRNAs can recognize that it's the correct amino acid to go in the next position when they come into that A site of the ribosome. We looked last time at the structure of a tRNA, right? So remember, a tRNA is a short RNA that folds up into a particular shape based upon its nucleotide sequence. So you get some hydrogen bonding here that kind of makes this clover leaf type shape. Importantly, there are 2 ends to it that are very important. This end over here, the 3' end of the RNA, remember, it's a nucleic acid. So just like DNA, there's going to be a 5' end and a 3' end, right? So in this case, the 5' end is over here, and you'd follow the strand around. It winds to the left and ends up at the 3' end. And remember, the 3' end has a free hydroxyl group on the ribose sugar, right? That's what makes it the 3' end. This hydroxyl group, this 3' end, is where an amino acid will attach to it. Okay. In this case, the amino acid that gets added is alanine. Right? And so alanine gets put on there through an ester bond, and that creates what we call an aminoacyl tRNA, right? Because on the left here is just the tRNA by itself floating around. Now it is coupled to and carrying an amino acid. So this molecule is technically different now, right? So we call this an aminoacyl tRNA, or more specifically, because it's an alanine, an alanyl tRNA. So a couple of things I want to point out to remind you. These cartoons are not drawn to scale, right? It looks like this amino acid's pretty big being carried around by this tRNA, but in reality, it's very tiny. Think about it this way. The amino acid, right, just is your typical, 3 carbons here, right? So you have your amine end, your carboxyl end, and the R group in this case is a methyl group. That's what makes this an alanine, right? Those carbons, oxygens, hydrogens, whatever, as an amino acid is attached to this adenine at the end of the tRNA. And think about the adenine. The adenine itself, right, is an adenine nitrogenous base, a ribose sugar, and a phosphate group, right? So the carbons, oxygens, hydrogens, and those in this cartoon should be just as big as the carbons, hydrogens, oxygens that you see in the amino acid here, right? So in reality, the scale should be that this amino acid is a teeny tiny thing on the end of this, all right? So why does this tRNA specifically carry an alanine? It's because the other end of the tRNA over here, the bottom of the cloverleaf, is what we call the anticodon, because 3 nucleotides in the anticodon in this position here, these are what have a chance to base pair with the codon sequence in the mRNA, right? So this tRNA is going to come into the ribosome in the A position, and because it fits in the ribosome in a certain way, this bottom part of the tRNA, the anticodon, has a chance to line up with the next 3 nucleotides of the mRNA that this ribosome is moving along and scanning, right? And so it's these 3 bases in the tRNA that have a chance to line up. If they don't line up properly, then this tRNA is just going to fall out of the ribosome, right? But if, let's say, a tRNA comes in and its 3 bases do match up with the next 3 bases in the mRNA, right, then you're in business. And so that's why the 3 bases in the mRNA, in this case, would code for, in this example, an alanine, right? So if you're looking on the table, the 3 bases would be an alanine. And then you'd realize that's why this tRNA that's carrying the alanine, it has the right anticodon to bring it in there. All right. So if you look at this, 2 of the 3 bases in the anticodon are very specific. In this case, it has to be in position 1, a C, in position 2, a G, right? In order for it to properly line up with whatever codon it is that's going to code for this alanine. But the 3rd position is what we call the wobble position, right? And just like the name suggests, you have some wiggle room here to make a little bit of sort of, you can sort of fudge a little bit of the interactions, right? Remember, our triplicate code is degenerate, right? Because with 3 bases coding for an amino acid, right? That's our triplicate code, right? You have 4 times 4 times 4, 64 different possible amino acids that you can code for, but we only need to code for 20. So that means several different combinations that vary by the 3rd position of the codon in the table, right, still code for the same thing. They would still code for alanine, even though the first 2 positions have to be a C and a G, right? And in this case, the nucleotide here is kind of a funny nucleotide. which we will talk about on the next slide and we'll look at here. All right. So the wobble position allows the third base in the codon to be a little messy, right? You can see here, here's the tRNA. It's trying to line up with the mRNA. In the mRNA, there's a G and a C. So the first two positions of the anticodon in the tRNA, they are in a position where they have to line up, right? So you get the proper base pairing of C across from G, C across from G. But remember, this is a three-dimensional molecule here, right? The way this is kind of like bending around at the bottom, this third position, you can see is kind of like away from the mRNA. So it's not actually lined up directly across. When you think of DNA, the two strands of DNA wrapped together, the bases are directly across from each other, right? So that they can form the proper, what we call Watson-Crick hydrogen bonding, right? 3 bonds between C's and G's, 2 hydrogen bonds between A's and T's. Right? But in this case, the tRNA, because it's bending away, you can have sort of like a weird angle of this third position. So let's look at this. So here you have a normal base pairing, the classic Watson and Crick base pairing. So if you have a cytosine across from a guanine, right, you'd have the 3 hydrogen bonds formed. But if the angle is kind of skewed, like if this were the third position, you could actually have a guanine in the tRNA at a funny angle across from a uracil. So instead of being straight across, right, they're at an angle, and at this angle, the guanine in the uracil could actually form 2 hydrogen bonds. It's pretty weak, right, but you will have interaction there. So that's why, in this case, a GCU, GC uracil, can code for an alanine, but so could a GCC, a GCA, or a GCG, right? Because that third position, even though it varies, because of this angle, you can still get away with some interactions, right? So that's the secret to how our degenerate code is read by the tRNAs. It just has to do with the three-dimensional shape of the tRNA and where that third position is allowed to have sort of a weird angle. So that means that in the anticodon, meaning right the loop of tRNA that's going to read the mRNA, this table shows you that depending upon which base is in the anticodon, right, these are the bases in the third position of the mRNA's codon that you can get away with actually still reading. So in this case, for example, this third position in this tRNA is an inosine, which is another purine. Remember, I told you there's like a bazillion different possible nitrogenous bases, right? So inosine is another purine that has been modified slightly. And so it can get away with reading a U, a C, or an A in that third position. So in the cartoon up here, it happens to be a U, right? So that means this tRNA, because it has a C and a G in the first two positions, right, it can read this as being an alanine, right? And other tRNAs that also have, let's say, other bases in this Walpole position, right, would also be able to read this as an alanine because of differences in that third position. All right, so here's a more realistic 3D view of what a tRNA molecule looks like. So even though for convenience sake, we draw it as a clover leaf, nice and flat on a piece of paper, hopefully by now, right, you guys all realize that molecules are never flat on paper like that. There's always going to be a three-dimensional shape in real life. So in real life, I like this figure because it takes the colors here, right, and tries to match them up with the colors where they would be once this thing is kind of folded up. And then you go ahead and you actually think about or imagine the electron orbitals around all of these things. And this, on the third part of the slide over here, this is the actual 3D shape of a tRNA. Okay? So again, one end is the anticodon end. So 3 of the bases here are the ones that are going to read or scan the mRNA. And if it matches, then this tRNA is going to then participate in the ribosome because at the other end of the tRNA up here is the 3' end, where the amino acid that it happens to be carrying, right, or that it is meant to carry, then is brought into the ribosome. So remember, the genes that encode for these things, right? Because after all, this is an RNA. That means it had to have been transcribed from a gene, in your DNA. Genes are just instructions for shapes. That's, I think, one of the simplest, but I think also most wonderful way of thinking about it. Genes, right, are just instructions for particular three-dimensional shapes. Without the right shape, this tRNA molecule might not be able to properly bind to an amino acid at one end, right? Or its anticodon part wouldn't be in the right position on the other end, right? It has to be exactly this shape in order to fit in the ribosome and do its job, right? And amazingly, this three-dimensional shape with all of this specificity is just coded for in the gene through a pattern of A, T, C's, and G's, right? I think that's pretty amazing. All right, so now that we've talked about this idea of how the tRNA comes in and how it's required to read the anticodon in order to actually stay in the ribosome and participate, let's look at what happens next, right? So this is now the process of translation. If we start off with the messenger RNA for a particular protein that's going to be made, again, right? So this is the final edited version of the primary transcript that was transcribed by RNA polymerase from the DNA sequence in the gene, right? And that took place in the nucleus of your cell. And then that primary transcript was edited, right? And you ended up with a finished RNA molecule, the messenger RNA, that then gets taken out of the nucleus. Remember, the mRNA has a 5' cap on one end and a poly A tail on the other end. These are certain ways of protecting this RNA molecule from being degraded by your cell, because otherwise it's just a random fragment of nucleic acid, and that might not be good, right? In fact, our cells, we evolved to not like just random fragments of RNA or DNA floating around in our cells. Why? Because that's what a virus looks like, right? Viral genomes, when they come into our cells, they don't have a 5' cap like this and a poly A tail, so they're targeted for degradation. But in this case, we want to preserve this because this is a message that's meant to go into the cytoplasm and be translated. Right? Part of the mRNA is not going to be translated, so that's the green part up here, but part of it will be, this middle part that starts from this green site. This green site would be the first position where an amino acid should start being put together to make a chain. And then this red site, this is the stop, right? This is where the chain of amino acids that's putting together will end, right? Everything in between is the coding sequence. So the three main steps of translation are initiation, so starting this, right, getting the ribosome to actually line up, come together and line up at that green spot. So the green spot is here. That's the first amino acid that's going to be translated. That's the codon for it, right? So notice it has little ticks in the RNA to remind you that each little tick here represents a different nucleic acid, right? So every three of them, 3 ticks, right, represents a codon, right? That's going to specify binding to the anticodon bases, the 3 little yellow ticks here that are part of this tRNA. And then the other end of the tRNA is carrying the amino acid. Right? Elongation is going to be how the process then proceeds. And the ribosome starts moving down the mRNA, and you have these different tRNAs coming in, now being moved through those 3 compartments, the A, the P, and the E inside the ribosome. In the process, right, you have a growing chain of amino acids coming out. And then by the time the ribosome reaches that little red spot at the end over here, that's the stop, right? The stop codon, or one of 3 possible stop codons from that table, right? Then the ribosome knows that's the end of putting together the polypeptide chain. Whatever that polypeptide chain is, it then comes off. And that's now well on its way to being folded up as a protein. So let's take a look at these 3 steps in a little bit more detail. So first off is starting the process of translation, initiation. Here you have the mRNA. Remember, on one end, you have the 5' cap that's kind of protecting it. You have a certain region that's not going to bother being translated. So it's just a whole bunch of nucleotides that eventually get to the AUG codon, right? And that's where the first amino acid is going to be. So a whole bunch of different proteins, these sort of blue, generically labeled 4G, right? So I'm not going to go through all the names of these because that's pretty advanced for this class, but at least know that 4G proteins are some of these proteins that start binding to the untranslated region. And remember how we said when we looked at DNA and how RNA polymerase II in the nucleus, when it's going to read the DNA to make an RNA transcript, it's brought there because of a whole bunch of transcription factor proteins, right, that assemble on the DNA based upon little sequences in the DNA that these proteins recognize, and then they pile up, and then the whole RNA polymerase II comes, is put into the right spot, and begins to transcribe the gene to make a big RNA coming off of it, right? Now that RNA has been edited, processed, and now it has moved out into the cytoplasm, where a similar mechanism is taking place, right? It's a similar theme. You have a long strand of nucleic acid, a whole bunch of proteins are going to assemble on it, and in the process, assemble the big complex of proteins that's now going to read that, right? But keep those two straight, because in the nucleus, what we're saying is you start with DNA, and so you have to pile up some proteins on it just to get the big RNA Pol II complex on it to read it, transcribe it into RNA. And now in the cytoplasm, you have the RNA that's going to have all these proteins assembling on it just to get the big ribosome on it. And that ribosome is now going to read that RNA and translate that into the finished protein product. Right. Because of that, this pile of different blue 4G proteins, that assembles the ribosome right at the right spot. So the 40S part of it first comes on, right? And then there's the very first tRNA that codes for, or that is hanging onto methionine, right? So that tRNA has the right anticodon to read the AUG codon in the mRNA. So this tRNA is positioned there, and therefore the first amino acid that starts off the chain is always methionine, right? Then the 60S part of the ribosome comes on, and the whole thing is assembled, right? Notice that when it's assembled, the very first tRNA is actually assembled with it already in the P position, right? So that the next tRNA that comes into the A position, right, this yellow tRNA that's in the P position can pass that methionine onto the next amino acid that's brought in. Right? So then elongation. You're going to have a whole bunch of tRNAs floating around in your cell like this one here. They're not just floating around by themselves. They're bound to proteins, proteins that then facilitate or provide a way for tRNAs to actually interact with the ribosome. In this case, they're bound to a group of different proteins known as elongation factors. Again, that's kind of a generic name because there's a whole bunch of proteins that serve as elongation factors. But you can see here, this EF1 is carrying around this orange tRNA. This brings it over here, right? Or more accurately, everything's just floating around. So by chance, it bumps into this position, and it puts the tRNA into the A position, right? Now, if the anticodon of this orange tRNA is not the right one to read the next three little pink bases of the RNA, this is not going to be very stable. And then this whole thing is just going to come off again, right? But if the anticodon of this orange tRNA that comes in does match up with the codon of the mRNA, this is stabilized. And then the EF1 protein, right, it's carrying around a molecule of GTP. It cleaves this GTP. This is guanosine triphosphate. It's another example of an energy molecule. It breaks one of the phosphates off and uses that energy, right, to encourage then the tRNAs to stay in position and start moving through the ribosome. So look at this. The yellow tRNA was the very first one, right? It carries this dark purple. This is methionine, right? Amino acid 1. And so when the second orange tRNA comes into the A position, is stabilized there because it properly matches the codon, right? And then the elongation factor protein shoves it over into the P position, right? The amino acid chain, that methionine, then gets added to the end of this next amino acid that comes in. And that's how the chain grows, right? And then that previous tRNA then exits. Okay, and this keeps going until the ribosome hits one of those codons that codes for stop. Right? So that's termination. So here you can see the ribosome has scanned all the way. So it started back here where this green region is, right? And we said this little green spot coded for methionine. That was the start of it all. Now this ribosome has worked its way all the way down the mRNA. Along the way, constantly tRNA is being brought in and the peptide chain growing here. Right? Eventually it hits one of the stop codons and there's a protein, a release factor protein that hangs out on the stop codon. And what that does is it basically then causes this process of translation to stop. Here's a nice illustration. Just like RNA polymerase II complexes reading down DNA, right? You can have a whole row of these RNA poly II complexes all in a line reading the same gene, making RNAs coming off of them, right? The mRNA that's being now translated in the cytoplasm can also have many ribosomes all reading it at the same time, right? We tend not to think about that, but yeah, that makes sense, right? Because why would you necessarily need to wait for a ribosome to get all the way down the end before another ribosome can start, right? That would not be very efficient if you need to be making a lot of this protein. So you can have a whole string or a whole line of ribosomes going down the same mRNA as you see here, right? And that means if you look at the ribosomes that are closer to the start, then the length of amino acid polypeptide chain that's coming out of it would be really short at the beginning, but it keeps getting longer and longer and longer as you follow that ribosome working its way down, right? Until eventually it hits the stop codon and that polypeptide is released, right? So here, I think this is cool, right? So this is an electron micrograph showing you can make out the actual strand of the mRNA that's being translated, and sure enough, a big bunch of these ribosomes working on it. So again, this gives you a sense of scale, because remember when we looked at an electron micrograph image that showed, let's say, one stretch of a DNA gene, right, and we had the RNA poly II complexes on it, you know, they weren't very big. They were like little black dots moving along, sort of a black line, right? Here, the ribosomes are huge. Right? You can really see that each one of these is a massive protein structure. All right. All right. Well, so, you know, when some of you students in the past have asked me, what's with the shape of this, you know, release factor protein? What is that? Right. You know, it's just, they try to draw it so it shows you they can bind to the codon, and then it also has some surface features that make it so that it can stop the process. But I remember when I looked at this, I thought it reminds me of something, you know, I couldn't tell what it was. And then I realized, oh yeah, it kind of looks like Charlie Brown, right? From the Great Pumpkin. Remember, he goes trick-or-treating with his friends and they all get candies and rocks and candies and cookies and things. And he always gets a rock, right? Anyhow, you don't have to memorize that because that's not going to serve you well beyond this class. All right. I think that description, hopefully you follow it, but it can be very confusing to imagine all this stuff happen. So here's an animation that I think suddenly makes it a lot easier. I found this just on Wikipedia, and I decided to use it because all of you can find Wikipedia, right? So that way you can watch this, because in the PDF version of the lecture slides, the animation doesn't work, okay? But in this slide, what they're showing is here's part of the ribosome. I think it's the 40S. And here's the mRNA. So this is all in the cytoplasm. And what it's going to show is how the ribosome gets assembled, the 60S and the 40S come together on the mRNA. And you'll see the process of tRNAs being brought in. So the tRNAs are represented like this. The dark blue is the tRNA itself. So on the end of it, you know, one of these little black dots, teeny tiny black dots, is the actual amino acid that it's carrying. And remember, it's being carried around by some elongation factor protein, the light blue thing, right? So together, the elongation factor protein is going to come in, help that tRNA try to fit in the A position of the assembled ribosome. Right? And if it works, great, because then you'll see the tRNA actually being involved through the 3 chambers of the ribosome moving around, and you'll see that the little black dots that the tRNAs are carrying are going to be put together into a growing polypeptide chain. All right. So keep in mind that that's what you're going to look for as I start this animation, because it goes pretty fast. Right. So here we go. So here it is. It's assembled, right? And you can see the tRNAs are being brought in by these elongation factors, right? And if it's the right tRNA, it'll fit. Sometimes it's the wrong tRNA and it'll just kind of like bounce off. I think it shows some of them bouncing off. Notice, right, the little black dots that are being carried in by the tRNA that's in the P position ends up transferring the growing chain of these black dots to the A position one. And that is your amino acid chain coming out. Okay, and it's emerging from the ribosome as a polypeptide chain. Okay, so I'm going to stop there with that one because it will then show some other functions that happen. But first, I want to talk about what those functions are. If you have this big, long polypeptide chain, right, and it's just a whole bunch of amino acid residues with different R groups, right? You have your big hydrophobic ones. Some of them are polar, some of them are non-polar, some of them are small. This is why you need to know your 20 amino acids, right? There's going to be a tendency, if you just had them as a chain, for them to fold up in many different ways. Remember my example of I have the little wooden blocks with little magnets on them, and there's many ways that I can encourage these side chains of these wooden blocks to interact, right? Certainly, not all of them, the resulting 3-dimensional shapes, would be the correct shape that this protein is meant to be, right? Because remember, it's all about shapes, right? So you have a challenge here. As this chain is coming out, right, it can start folding up on itself, right? Parts of the polypeptide chain can just start folding on itself before the rest of it has even come out, right? So that may actually mean that the shapes that you get are not the right shapes. So we have certain proteins that function to sort of chaperone this process, right? Or help out the process of proper polypeptide chain folding into desired 3-dimensional shapes. And so these chaperone proteins are often referred to as heat shock proteins. Okay, let's take a look at these. So here is a newly made protein. The purple thing is the chain of amino acids coming out of the ribosome as the ribosome is moving down the mRNA. Right away, certain heat shock proteins like HSP40 and 70 start binding to this chain. And what they're doing is they're trying to discourage the chain from starting to fold up on itself, right? Because you don't want it to really start folding until the whole chain is made, and then you have the opportunity to make sure that all parts of it are folding up the right way, right? So you can see HSP40 and 70, right? They're binding to it. Right? These have enzymatic functions. They bind to ATP, and when they break ATP, breaking off a phosphate bond from adenosine triphosphate, that gives them the source of energy, right? By a high energy bond being broken, those electrons want to do something else, something that's better, right? And so the newer lower energy bonds cause the chaperone proteins to move around. And by them moving around, it encourages the polypeptide chain that they're hanging onto to also be moved around, right? So they help properly fold proteins that way. Other heat shock proteins are much more impressive. So HSP60, for example, is a big heat shock protein complex that's shaped like a barrel. If you have proteins that are denatured, right, they can get fed into the barrel, and inside the barrel, you have ATP activity that folds the proteins up again, and then the properly folded protein is released. Why are these called heat shock proteins? What a funny name, right? I mean, chaperone is already funny, but heat shock protein is kind of weird. These were discovered when researchers would take cells, right, and expose them to a shock of heat, right? And remember, heat causes proteins to denature, right? 3-dimensional molecules tend to unravel if you heat them up, right? And so what they realized is when that happened, boy, the cell's in trouble because a lot of its important proteins are now unfolded. They're not going to work. They saw that the cell starts making a whole bunch of new proteins, once they've cooled things down, and those new proteins were helping the denatured linearized proteins get folded up again, right? So in response to a heat shock, these particular proteins like HSP 70, 40, 60 are made by the cell to try to fix things. So next lecture, we'll talk more about how the cell properly encourages the proper folding. Because even looking at this, you're probably wondering, yeah, but this isn't very specific, right? How does the cell actually control the proper folding? We'll talk about that next lecture. But I wanted to end up talking about mutations. So if you have alterations in the original DNA sequence for a gene, that could have certain ramifications or consequences having to do with once that genetic sequence is transcribed and then translated into a finished polypeptide chain. For example, in this figure here, on the left column is just, for an example, the normal sequence that should be found to illustrate an example here. So in this case, let's say the DNA is supposed to be GAA, at this part of this gene. So on one strand, it's GAA. On the other strand, of course, it's CTT. Maybe you have a mutation where that A in the middle changes into a T, right? So maybe this happened, for example, when DNA polymerase was trying to copy this gene and it goofed and it put a T there, right? Where there should have been an A. As a result, the codon in the mRNA should be a GAA, right? But it is now a GUA. As a result, if you looked this up on the codon table, what should have been this glutamine, right, as part of the polypeptide chain that the ribosome is putting together, now it's a valine at that position. Different amino acid, very different side chain on that amino acid. So as a result, that part of the polypeptide chain that's made may not fold up the right way, and the three-dimensional shape of the finished protein might be altered. It might be a subtle alteration. It may be a dramatic alteration, right? But it would impact the ability of that protein to function. So that's what we call a missense. One amino acid ends up substituting for another. Nonsense is if you have a single DNA base that you goof and you make a mistake and you change it, and that results in a stop codon where there should not be a stop codon, right? So the ribosome that's translating this mRNA, it's going along, blah, 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 blah. At this point here, it's supposed to then, have a tRNA that's carrying a leucine that comes into the ribosome, and the leucine then becomes the next thing on the end of the chain. But instead, because of this mutation, the ribosome hits a stop, right? Maybe it was only halfway through making the entire polypeptide chain, and as a result now, the polypeptide chain that comes out of it, right, is truncated. So again, it's probably not going to fold up the right way that the protein is meant to, or maybe even more dangerously, maybe because it's missing part of the protein, it's going to behave in a way that actually causes trouble inside of the cell. Then you also have silent mutations, where if you swap the third position of the codon, because of our redundancy, our degenerate code, what was a proline before at the end still is a proline. And lastly, this type of mutation, where instead of just mistakenly having a different base in there, you can sometimes have a mistake where you have an extra base put in there. So here's the wild type sequence. It should be ATG, AAG. But what has happened is somehow an extra C has been inserted between the ATG and the AAG, right? So what that means is the reading frame. Remember, it's supposed to be every 3 bases are read as a codon, right? But these bases are just a continuous strand. So it has to do with the fact that in the ribosome, every 3 bases lines up with one of those chambers of the ribosome inside where tRNA can match up. So what should have been methionine, lysine, phenylalanine, etc., right? This lysine should be coded by this AAG, but now you have this extra C. So the reading frame gets messed up. The AUG is still correct, but now the next 3 is a CAA, and that gets translated, right, into this glutamine over here, right? And then everything downstream from it is messed up also. So now you have a valine, and then it hits a stop, right? So that's really bad. Okay, let's go ahead and stop there today. There's one more slide in the lecture, but I'll save that for the start of our next lecture on Friday.